thought we could shortcut that, but I guess not. Does it look okay now?
You've got it.
All right, thank you. Well, I'm really excited to be here and to get to talk a little bit about what we do at UC Davis and in the field. So I think everybody here knows that the fields of stem cells, immunotherapy, gene editing, and regenerative medicine are really changing healthcare. I used to say they're poised to change healthcare, already changing healthcare. And of course, these are living medicines, so they're complex products, as we all know. They're not pills, and they require especially, they can't just sit on a shelf. They're frozen, they're fresh, they're manufactured. And we really need specialized facilities and a high degree of teamwork and expertise to manufacture, formulate, and deliver these new living medicines. And so that's what I'm going to talk about today, really taking these therapies from the bench to the bedside. So we're very fortunate here in our stem cell program, first of all, to be in California. So we have funding from the California Institute for Regenerative Medicine, NIH private donors, California state and foundations. We do a lot of team training. We have team training initiatives, as you do, and a large GMP facility. So currently, we have 48 cell and or gene therapy clinical trials ongoing, over 20 in the pipeline, and we meet based on different teams and try to move the ideas for the products from the bench to the bedside. So this is what I do all day, is meet with teams. We meet with the FDA. We prepare the paperwork, and we try to move all of these forward. Some are industry sponsored trials and some are investigator initiated trials. So I'll go through just a couple of those and how we get there. We have this great GMP facility. It's 6 individual suites. This is available cellular product manufacturing for clinical trials that have US FDA approvals only. It's very large, and we're manufacturing for many academic and industry partners, and of course throughout the UC system, and many academic collaborators as well as industry partners. Largely the startups will come to us. Everything has a standard operating procedure. It doesn't exist if it does not have a standard operating procedure. We start those in the lab, we do them in the lab, and then we transition to the GMP facility. So really going toward FDA approval, we start with the initial efficacy data, initial safety data. This is in the lab. We start with a pre-IND meeting if the FDA will agree to have one with us. Often we'll submit a brief package for a pre-IND meeting, and they will send back some written advice. We then go into further proof of efficacy and safety, including the plans for the pivotal studies that come later. We'll draft the clinical protocol and the monitoring plan, and of course the standard operating procedures for the CMC sections, the chemical manufacturing and controls section. And we'll put those into the pre-IND package. The FDA will have a pre-IND meeting with us. This is a really important step in the product's lifetime, because this is where the FDA will give you the specific feedback that you really need to follow when going into the IND package. So for the IND, you need the pivotal safety studies to be done, finalized clinical protocol and monitoring plan. And you have to have made a batch of the product, because that's where you get all the details for the CMC section, and you have to submit the details, the batch records need to be done, and the certification of your product need to go into this IND application. If that clears, you can start the clinical trial. So this is another way to look at it. It's development, preclinical. You can, if you clear that, the IND, get the IND approved, you can go into phase 1 through phase 3, and then the BLA is submitted. This is usually after what I do. I'm purely academic. We work with some companies, but I have never submitted a BLA, full disclosure. We very much work in this area here. And through it all, safety and quality need to be done. The principles of GMP need to apply throughout the manufacturing. We start the batches of mesenchymal stem, stromal cells. We start them in the lab as a tox batch. We do some of the animal testing, and then we do the bridging studies with the product that's manufactured in the GMP facility. So that saves us some time. we're using the Terumo Quantum device to make lentiviral vectors for gene therapy and also for our MSCs now. This is a hollow fiber device. We really like it because it's 3D and cells like the packaging cells and the MSCs really like to be in this environment rather than flat on the plastic. And currently we have 19 of these in our GMP facility. So I guess we like them. We're going into a phase 3 clinical trial for a corporate partner, and about half of these belong to them. So 19. So just to step back just a little bit, for gene therapy, which is all the rage now and is actually being successful in many diseases, viral vectors will carry the gene of interest to the patient. So for lentiviral vectors, the cells are removed from the patient and modified ex vivo in a GMP facility and then returned to that patient. AAV vectors, adeno-associated virus vectors, can be delivered directly into the patient, into the bloodstream or a target tissue like the brain. And this is another way to look at it. The direct delivery, AAV, would be here versus the cell-based delivery, where the therapeutic transgene is packaged into a delivery vehicle, such as a virus, by a packaging line. And then the therapeutic transgene is introduced into the delivery cell, such as a stem cell, often derived from the patient. And then these gene-modified stem cells are re-administered. So we're doing several trials with this. I'm really not going to talk about pluripotent cells today. We don't use them routinely. We have a few trials that are ongoing with them. The risk for a GMP facility with these, quite frankly, is that they really like to duplicate chromosomes. So you'll get to the end of a really expensive batch of cells and find that there's karyotypic instability, and then you have to scrap the entire lot. So that's a lot failure. So we really have been focusing, most of those clinical trials that I showed you, except for maybe 5, are with somatic or tissue-specific cells. So I'll talk about hematopoietic and mesenchymal cells today. So first of all, the blood-forming cells, hematopoietic, they're in bone marrow, cord blood cells collected after birth, or mobilized cells that come out of the bone marrow into the blood with a mobilization agent. They can make every cell in the blood system, including the immune system. And if you can genetically engineer this cell, you will have a cure. You should have a cure for the lifetime of the patient, because upon infusion intravenously, they'll go back into the bone marrow. Always fascinating to me how they home back into the bone marrow, and they'll set up residence for life. So this is what the hematopoietic stem cell gene therapy looks like. Cells are collected from the patient. The blood stem cells are selected and purified. We have machines that do this in the GMP facility. And then a working copy of the gene is inserted into the cells using a viral vector. The gene corrected cells are frozen or cryopreserved. And then the patient will be conditioned to receive the treatment. There needs to be some conditioning to make some space in the bone marrow, either space or to set up some kind of damage, whereas chemokines are calling the HSCs in from the circulation and having them lodge, extravasate, stick, and lodge back into the bone marrow. And this slide is from Dr. Don Cohn, who I'll mention in a moment, and he has shown that the conditioning is very important, even if it's the patient's own bone marrow stem cells. So then the gene-corrected cells are infused, and the patient is monitored on the trial. I started, Dr. Lane mentioned how long I've been in this field. I started 30 years ago with Dr. Don Cohn, and we started working on hematopoietic stem cell gene therapy for babies with bubble baby disease or adenosine deaminase deficiency, and taking their own cord blood cells, engineering them with a good copy of the ADA gene, and returning to the patient. I went on for my career after 15 years training with Don, and then his team kept at it and started conditioning the children or babies who were receiving this autologous, their own bone marrow stem cell gene therapy. And so 30 years in total, his team kept working on this, and now they have actually functionally rescued, functionally cured 50 children born with this type of primary immunodeficiency, ADA deficiency. And so this is the paper that they had just recently in New England Journal of Medicine, autologous ex vivo lentiviral gene therapy for ADA deficiency. And this is showing the patients from Dawn's group in blue, the US studies, and from a collaborator in the UK in the green. And this is patients without an event. And so these patients literally are cured, they're not on medication. And he just did there was a further report following the first 10 patients, and for one of them, it wasn't so successful. He didn't have the right conditioning, but for 9 of them, for 10 years, they've been stable and off of all medication. So Dawn just got the International Society for Cell Therapy Career Achievement Award, and well-deserved. Now, I've been working more with the mesenchymal stem, stromal cells, whatever you want to call them. We initially called them mesenchymal stromal cells, and then it was discovered that they could make several lineages of cells, and so it became very popular to call them mesenchymal stem cells, so we switched, and now it's back to stromal for some reason, but I still call them MSCs. I usually say mesenchymal stem / stromal cells, MSC, and then it's MSC for the rest of the grant or whatever, and people can argue as much as they want. But so these are adult stem cells. They can't create an entire tissue like an embryonic or induced pluripotent stem cell might be able to, but as I think everybody on here knows, they can repair damaged tissues by responding to the scene of the injury and producing healing factors. They're the paramedics of the body, and they have a very strong safety profile in clinical trials, although large phase efficacy has yet to be shown convincingly in large phase clinical trials. What the biology of these cells never ceases to fascinate me. So this is MSCs in red and damaged muscle fibers in green. The MSCs communicate with other cells by producing growth factors, by actually touching them to directly infuse factors, and also by sending out microparticles and exosomes for other cells to pick up. And I find that just so interesting. So in this video, you can see, again, the MSCs are labeled with a lentiviral vector with tomato red. And by the way, we have a vector core if you need to do this or need help with transducing cells. You can see that they're putting out these little microparticles, and that in some cases, the green muscle fibers are taking them in and just absorbing them. We can show that microRNA, protein, and even organelles in these microparticles and exosomes that are coming from the MSCs are successfully transferring into the target cell. And I find that just really interesting. This is showing actual mitochondria transferring down nanotubules, and nanotubules are of course like a kind of opening a gas hose into the other cell, and cytoplasm, things in the cytoplasm can transfer between the cells, although things from the nucleus rarely do, the DNA rarely does. So we have been studying exosomes as well, and Dr. Jonathan Anderson has been the lead on these studies, and we put out a paper a few years ago, comprehensive proteomic analysis of MSC exosomes reveals modulation of angiogenesis via NF-kappa B signaling. And this was looking at the proteome of the MSC-derived exosomes and showing that how they contribute to angiogenesis. So I mentioned, many clinical trials are ongoing with MSCs, as we all know, for tissue repair and revascularization, modulation of the immune system in graft versus host disease and organ transplant, enhancement of hematopoietic stem cell engraftment through co-transplantation, and also reduction of inflammation. These are the largest ones, and also a lot of tissue engineering clinical trials ongoing now. And of course, the MSCs are not becoming the damaged tissue, but acting through their secretome and the microparticles and exosomes that they secrete. So we've worked for several years now on mesenchymal stem cells overexpressing VEGF to treat critical limb ischemia. We are still working on this. We are at the IND stage and ready to complete the pivotal safety studies. We had CIRM funding, and as CIRM was winding down, the CIRM funding dried up, and so we are moving forward again with this this clinical protocol. And I just wanted to mention the formulation. So formulation is really important for hematopoietic stem cells. They can be frozen for 20 years. They're in DMSO. You take them out of the freezer. you thaw them bedside and you put them in the patient's vein. And if the patient has had, as I'd mentioned, something done to their bone marrow to call those cells in, they will engraft. MSCs don't really love to be frozen and handled like that. They're big cells, they're adhesion dependent, they like to engage their integrins. They don't really like sitting around in bags after they've been thawed for hours until they can be administered. And we are working on formulating them so that they're cryo recovered and they really get everything together. I could go into this for another half hour, but I won't. We are doing a cryo recovery on demand formulation phase. We're thawing and, thawing, oh, going the wrong way. Sorry. I will get there. Not sure why it went to the end. Almost done. We're thawing and preparing them in the GMP facility and doing the final formulation, and then planning to ship them in formulated syringes ready to inject. So that's just really to get to our catchment area with the distant hospitals. These cells will be injected into the muscle for the patient, into the leg muscle where the ischemia is happening for critical limb ischemia. So the formulation is really an important thing to think about. I wanted to mention CAR T cell therapy for cancer because the manufacturing, the formulation, and the cost are all really, really important. And that's something that our group is doing a lot right now. And it applies to every product that we're manufacturing, but this is just a really good example. So CAR T cell therapy is cell-based immunotherapy. It uses gene-modified cells from a cancer patient's own immune system, and it can give cancer patients another chance at life after relapse. So the gene-modified chimeric antigen receptor, or CAR T cells, can seek out and eliminate cancer cells throughout the body. So there are 2 products approved for liquid cancers and a lot of clinical trials ongoing for solid cancers for tumors. This is what it looks like in our facility from blood to investigational product. The problem with these, the commercialized therapies, those that have been through the BLA, they're FDA approved, they can be prescribed by the doctors. The list prices are $500,000 and $373,000. The CMS reimburses the hospital at 65% of estimated costs. And so really the costs need to come down and need to be in line with what insurance will fully reimburse so that we can provide life-saving therapy to all patients in an equitable manner. Because you could imagine someone with private insurance where it's completely covered is less of an issue than 65% covered by CMS, where the hospital is losing, $150,000 per patient per treatment, and that's just for the product. That's just for the product, not for the care of the patient. So we're really working on this and working on ways to deliver these immunotherapies. And for any of these therapies, and really important thing to think about is cost of goods. This is the inner workings of one of those Terumo devices, the cartridge and all the tubing. And, it's complicated, it's expensive, it's several $1,000s, and it's very hard to get that down, but we can automate things. We can reduce the labor cost, we can streamline things more and really bring down the cost of goods. So one way that CIRM is helping us do that is by sponsoring these Alpha Clinics, the Alpha Clinics Network. There are 5 sites, City of Hope and UCLA are together in this down in Los Angeles area. 5 sites with these clinics, and the goal is to deliver high quality stem cell clinical trials to patients. So we're service funding the people for the infrastructure to do these trials. And so we have the Alpha Stem Cell Clinic clinical trial unit. We help with study initiation, regulatory IRB, budgeting, contracting, CRC, the clinical research coordinator, and have a cell infusion nurse practitioner. And so the GMP and quality control testing lab is doing the manufacturing, and they're manufacturing the cell and gene therapy products using SOPs. They test and cryopreserve, they do the batch records, they perform the lot testing, sterility and potency assays, they document and complete the certification. And then the Alpha Stem Cell Clinic will take over at that point. do thawing and formulation of these products, perform the release criteria testing, sign off the product, and deliver it to the clinic using the STEM mobile. So this is just a list of the things that you don't even think about that need to be done, but for these living medicines, these things are really important. It's not just, you know, a dry vial that can be shipped and reconstituted. It's cells or vectors that need to be handled correctly for administration to the patients. We can't kill the product during the formulation. And so one of the greatest ones that we've been doing just recently, it started in this July, was Dr. Diana Farmer's treatment for spina bifida in utero. And so spina bifida is where the nerves are, you know, they haven't, the notochord hasn't closed properly, and the nerves are sticking out of the baby's back and getting damaged, and it causes paralysis. They're going in utero and doing the surgical correction. They have placental MSCs on a matrix that they're covering the surgical wound before they close the skin. And the first baby was treated and has been born, and we're waiting to make announcements on how that's going for first follow-up. And the second patient is scheduled to be pretty soon. So we've been with them the whole way, you know, doing the preclinical studies, developing the SOPs, developing the placental stem cell banks in our GMP facility, going through the FDA, going through all of the CIRM audits, and just very, very happy that this clinical trial has started. And they had a lamb model of spina bifida and also a naturally occurring bulldog model for many years. So it's very, very gratifying for me as a PhD to get to the end where the patients are being treated and seeing that these therapies can impact lives. So last slide, in this new era of living medicine, cell and gene therapies are being prescribed to treat diseases and injuries. This is a list of those that can be prescribed, all very expensive. We are very committed to training the healthcare providers of the future to deliver these living medicines, and we're also committed to bringing down, helping the field to streamline and bring down the cost so that they can be delivered in a very equitable manner. This is our team. It really takes a village to formulate and deliver these cells and manufacture them. And you can find me on Twitter or my email. Feel free to reach out. And so that is all I have today, and I would be delighted to take questions if anybody has some.
Yeah, thank you so much, Jan, and for showing what a great group and facility you've been able to put together in Marshall through, especially these through the pandemic times, and keep going. Jan's got a couple minutes for questions. Go ahead, and anyone can ask them.
I went super fast. If not, we can just go on to Jim and see if anybody has some at the end. That's fine.
Okay, all right.
Thanks, everybody.
Oh, wait, here we go.
Have you in the chat? There you go.
Yeah, there's one in the chat right now, Jan. Can you see it?
I can see it, yeah. Interestingly, we don't have a clinical trial manufacturing keratinocytes, but I don't see why it wouldn't work. Any cell that is adhesion dependent should do well in that device. But I don't have direct experience in that. Good question.
May I ask another question? This is Isabel. Yeah. Hi, Jen. Thank you for that nice overview. I'm interested in seeing what your experience has been in comparing cell therapy with gene therapy. Did you encounter the same problems there? I would think that there are different things that you should often think about, and that the cell therapy is more complex than gene therapy, but I'd like to have your opinion about that.
Yeah, they are very different. You know, everything from how we manufacture and handle the two products to the outcomes. The AAV vectors have been having some great successes in some CNS diseases where it's hard to transplant the cells. There are certain capsid proteins for AAV that seem to be doing well. They both can have their problems. The lentiviral vectors need to be, integrated into the cells, and the AAV can be delivered into the bloodstream or into the CNS for the brain. For the gene-modified stem cells, FDA needs us to have an average of 1 integrant per cell. They don't want us to just barrage the cells with lentiviral vector. And so you need to be very careful with your multiplicity of infection and get the product just right. And so that's very complicated, and it has to be very standardized. And they don't want us peppering the genome with lentiviral vectors because they're integrating randomly, and that increases the chance for oncogenesis. As for the outcomes, they're really different. The best outcomes so far have been for the hematopoietic stem cell gene therapy, and that's why I wanted to mention that. Don's really the pioneer in the field, but there have been several other groups that have had long-term enduring and safe hematopoietic stem cell gene therapy. And so I think, for that field, the HSCs are really leading the charge. But I will never give up on using MSCs to deliver gene products, at least for a few weeks or a month, to the local tissue that needs it. So that's really my passion. We shall see. It's been before its time for, 5-10 years. Wrote a book on it 10 years ago.
Sure. Thank you.
Thanks for that question.
Okay, very good. So thank you so much, Jan. And now we will move on to the next talk, which is going to be given by James Kovac, who, as I mentioned, is the Chief Innovation Officer and really is working to commercialize university inventions, which is what we all like and want. So please, Jim, looking forward to your talk. Thank you so much.
Thank you. Can you see my slides?
Yes, yes.
All right. So yeah, just to be synergistic with Jan's talk, I wanted to talk about kind of where she leaves off. Remember, she said in that time course where she does a preclinical, then the clinical trials, and then she basically said, universities, health systems don't file BLAs, the biologics license application that allows a therapy to be put into interstate commerce. Typically that's done through industry partners. And so I wanted to talk about how how the university academic tech transfer has basically gone from kind of its start to a really highly industrialized and interactive agency with industry to basically bring these therapies to market, as expensive as they are. But nonetheless, I wanted to just talk briefly about where the history of it is very interesting to me. It's got a strong California component to it as well. The Big Bang, what I call 1980, that where several things came into being at the same time to really kind of give rise to tech transfer. And then talk about some of the steps using Jan's own lab as an example of how the system works. So just to mention that really there was no university industry relationships sponsored by NIH prior to World War II, where Vannevar Bush wrote a memo or a kind of more than that, a report to President Roosevelt, essentially how research and innovation helped win the war. And that translated into newfound efforts of the federal government to basically conduct research, not only through government labs, but through universities. So it was the starting point of how universities now are kind of the cultivation of innovation and the handoff to industry. Just to look at the NIH budget in 1938 was $500,000, and currently it's $43 billion. And an additional $12 billion in national laboratories, so $55 billion in federal funding. And so, I think the federal government has seen the rapid increase in patent applications, because this is early stage innovation where, as we'll talk a little bit more about the requirements of a patent, we're developing patentable subject matter. And so the question is what to do with that. And I think the federal government wisely saw that it's not very efficient at really commercializing or helping to commercialize intellectual property. So basically, one of the elements of the Big Bang in 1980 was to create the Bayh-Dole Act that provides the ability of universities to take title to government-sponsored research, which is, you can say in a sentence, but it's really had a revolutionary impact on innovation in the U.S. There were certain aspects of the Bayh-Dole Act that we'll talk about, but I just did want to mention the other elements of the Big Bang, I call it, is when a lot was going on at Stanford at that time. Stan Cohen was there. Herb Boyer, of course, was at UCSF. Paul Berg won the Nobel for DNA sequencing in '80. The Bayh-Dole Act was passed, and we're going to mention that in the rise of tech transfer. Amgen was founded, it cloned and patented the EPO gene itself, and then Diamond versus Chakrabarty was a patent case that basically said all things under the sun are patentable. It can't be a natural sort of formula or rule of nature, but it really is a broad mandate to enhance and accelerate patenting, and a lot of that was going to occur at universities. So the Bayh-Dole Act said the federal government now is saying the university will take title of inventions from federal funding with some caveats. Basically, the university has to act quickly. It has 2 months to make the decision to patent. It has to establish tech transfer offices, and we've seen these throughout the country in all the major institutions. It has to take efforts to actively commercialize the invention. And in certain aspects, the government actually may have walk-in rights, national emergencies, and if there's undue foreign influence over patents, the government does retain certain aspects of rights. And so the university has to basically say, okay, we're going to move forward with this. We give the government a license, non-exclusive and only within the federal government to use. It doesn't really have an impact on licensing to startups or to existing companies. But there is a small business priority that universities must give, which is really kind of help facilitate startups around the country. And any product has to be manufactured in the United States. And then the two last ones, excess revenue must go back into research and education, and the university has to share royalties with inventors, which is proven, I think, to be a significant incentive for, at some level, for faculty members to basically pursue, licensing. So I just wanted to say a couple of words about what it is that we're licensing. What is intellectual property? I mean, this is certainly a broad area, but I think there's a couple of teaching points or learning points that I always think about. One is, basically the four elements of intellectual property are generally regarded as patents, copyrights, trademarks, and trade secrets. So a trade secret has its value because by definition, it's not disclosed. And so it's kind of the antithesis of university-based research. And we don't really deal in the universities with trade secrets. Rather, copyrights, worth mentioning that software is considered copyrightable. So, there's some element of innovation in universities with copyright. Trademark, generally not something in the research realm. It's really patents and the rights of a patent to make, use, or sell that are relevant to the sponsored research from NIH or a company. So when does an invention essentially spring into life? It's really at conception. So if an individual faculty member has formed a complete and operative idea, you don't have to have reduced it to practice. You don't have to have showed that it's going to work. You just have to put it down on paper, and that's when you've conceived of it, that someone else could actually take those directions and follow them. The reduction to practice typically occurs contiguously, but doesn't have to. It's really the actual successful construction of the device itself. And so patents have traditionally been described as, as an analogy would be to real property. I mean, the right of real property is the right to exclude. I'm on my property, you can't come here. So that defines your property, the area. It's meets and bounds, basically, back in the hundreds of years ago is how it was described in all the way back to England. So it's the same elements of your invention gives you the right to exclude any other party from making that invention. Doesn't give you the right to practice your invention. And that's a very important point, because you could invent, the inventor of a bicycle basically has the right to build the bicycle. The inventor of a wheel, right? A particular wheel doesn't necessarily have the right to make a bicycle. He's got to get a license from the owner. And if it's the bike owner of has really wants a specific wheel that's separately patented, he'd need to actually work with that individual, because he wouldn't have the right to build the bicycle with the patented wheel. So it's really kind of interesting that freedom to operate, which is a subject of its own, is something that we think a lot about at universities, but it's typically a very sophisticated, review, and one that is typically not done in university. We really kind of focus on, is something patentable? And if it is, the right to exclude, and it can be sold, assigned, licensed, so we can basically do a deal around the patent that has been filed on. And then the term of patents currently is 20 years from the date of filing. Here are the things that are patentable. It's basically composition of matter and methodologies. So articles of manufacture or compositions, processes are a whole separate category, but it's basically methods and compositions. Laws of nature are not patentable, but in that Chakrabarty case, because the DNA in terms of was isolated, was taken out of the natural confines, and then incorporated into a bacteria. And actually, was the dispute was around a bacteria that had incorporated a oil ingesting enzyme. So which you don't see around, we don't actually see that in practice. So it's another kind of point, is that a patent doesn't have to actually work or be functional in order to be patentable. But novel, useful, and non-obvious, these are the three requirements for a patent. Can't have been known by others or used, which is a big bar, because in today's day and age, I mean, if you have a great idea, there's likely others who are at least thinking about it. And so it's got to be useful, which is typically not a big bar in the work we do in academia. And it also has to be non-obvious, which is the one that is most litigated and most kind of fought over in patent offices. Novelty is identified by doing massive novelty searches, and that's what we do at UC Davis and other academic tech transfers, and also the federal government has patent agents who do the same thing. And usually you can identify if something's novel or not. But the non-obvious is would someone skilled in the area be able to logically deduce from things that were known? Now that's a very complex and kind of subject to human interpretation sort of bar, and is the one that we spend the most time with patent agents in the federal government trying to get on the same page with. And so inventors are any intellectual contributor to the conception of the claims of the patent. It can't be someone who just works really hard 24 hours a day, but is told everything to do. I mean, it's got to be participation in the conception. And then ownership follows inventorship. So the owner originally is the inventor. But in the legal environment, I'm going to skip ahead here and then go back. In universities, faculty members in their contracts, and you see the university kind of form here have patent acknowledgements where faculty members assigned to the university then take over the commercial with the licensing of that. And in cases where there's two inventors, say one at UC Davis and one in, say, Baylor, I'm just kind of picking one out of the hat here, is typically, if it's a co-invention and you want to license it, the two universities will agree on one university kind of taking the lead in terms of interacting with industry, but keeping the company closely, they're keeping the other university kind of in the mix, but not wanting to confuse the market. You know, that Cohen-Boyne patent, which was for recombinant DNA, they decided early on that Stanford would take the lead in that. And it resulted in you know, a historic deal and really kind of one of the elements of forming Silicon Valley and the biotech industry itself. It's interesting that I think we talk about it later, but you can, or maybe we have already, but you can either, a tech transfer office can either choose to license something exclusively or non-exclusively. And Genentech at the time, one of the first biotech, said, hey, we would like an exclusive license to recombinant DNA. And you can imagine how different the world would be if Stanford would have done that. They actually considered it, but they thought it would be it's a broad-based technology, one that was better used broadly, and they adopted a non-exclusive license program that ended up licensing nearly 1,000 different companies. So that was obviously the right thing to do in retrospect, but an example of how you can actually kind of set your strategy, exclusive versus non-exclusive. And here's a screenshot from the USPTO. I encourage everyone to go there. There it's up top, uspto.gov. It's great for learning and for searching. And lo and behold, here's Dr. Nolte's invention with the co-inventors, a brief description, the patent number over on the right, the date of the patent itself. And it's there for everyone to see, because that's the deal. You get a patent, you get 20 years of exclusivity, but then in exchange, you have to tell people exactly what you did, and you have to teach them how to make it so that they can make something yet better in the future. And just another element we won't have time to go into, but here's an example of the metes and bounds. This is the claims, which give the breadth of the patent itself, and which set the standard of what UC Davis could prevent others from practicing. So it's not the right to practice this, it's the right to prevent others from practicing it. And so one last thing on joint ownership here, I think I got a minute or so left, is so Cohen-Boyer, or any patents that are developed by multiple individuals at different institutions would make that a joint ownership. So any joint owner can license or assign the full patent itself, which is typically, again, why the universities go to such lengths to create kind of a uniform front and basically have a single entity be in charge of the licensing itself, because it's just a better way to transfer to industry if you don't have competing interests like that. It's interesting to look at CRISPR, the CRISPR case right now that involves two separate universities, not the same patent. So there's been lawsuits and it'll work itself out, but between UC Berkeley and MIT Whitehead, I think, relating to CRISPR-Cas. And there's not the same invention. So there was not the same, kind of interest or logic in the parties working together. And indeed, they've not worked together. But, like I said, the business environment has a way of basically working things out in a way that keeps these products going to market. Meaning, CRISPR-Cas, there's certainly not been a lack of investment because there's not a perfect, agreement between the two academic institutions that have kind of these core patents. And it's just a it's a statement that, you know, life goes on. These patents are really important, but there's always more innovation. That's the beauty of the system, is that it works to help companies, but then it also sets the stage for additional innovation. So you have to apply before the main one is before you describe the patent or publish on it. And that leads oftentimes, since many times inventors it'll occur to them, hey, maybe I should talk to my tech transfer office, you know, a week before a presentation or a publication or something like that. So that does happen, and it's something that hopefully this group, if you think of an invention, you've got to go early to your tech transfer office, because those fire drills, as we call them, they're never fun to actually go through. So although in the US only, if you miss a deadline, you publish, you still have a grace period for a year for just a US application. And not that that's optimal, because you like to think about having worldwide rights, but it's just something to mention. And then if you look at the cost, it's really, in the grand scheme of things, it's not that expensive to get a US patent. Really what happens is when you want to have a worldwide patent estate, you have to start filing in individual companies about a year and a half after you file your US application. That's when the expense really gets high. So by that time, you really want to have a license. And so what we do in tech transfer is kind of distinguish between the patentability that I've looked at, but then the marketability is essentially not only how is this going to fit into a product, but then who's the right licensee? Is it something that's like a platform that we can spin into a company, and is the faculty member interested in that? Or is it best to just try to identify one or multiple licensees? And when we license, there's that question, do you do it exclusive or non-exclusive? In concept, you could do fields of use and territory and kind of divide up your patent in different mechanisms to really kind of capture industry, but also to get a return on investment. So yeah, I've got a couple of minutes for questions as well. So my closing statement is to, I'm looking at the tech transfer offices of this organization. I think I saw there's USC, but then also a reference to Michigan and University of Pittsburgh. And these are great and well-known tech transfer offices. And tech transfer individuals love to hear from faculty and love to kind of talk about what they're doing. And I would encourage people to reach out for additional information. And so I'm open for any questions that anyone had, but thanks for allowing me to give a presentation.
Well, thank you, Jim. That was very informative and very useful to our entrepreneurs on the call. So now your talk is open for any questions.
I wanted to comment, you gave the exact example of the basically genetic engineering all potentially going to Genentech, and that it would have clearly been less beneficial to the public if that one company had all that technology that they would not be able to as effectively get out.
Right.
Bayh-Dole has an aspect that there's an obligation in the licensing to get the technology into the hands of an entity that's going to do the best job at getting it out to the public. And we encountered this with one of our circulatory support devices, where basically a company had licensed the technology but was sitting on it because they didn't want it to compete with their technology. But it was hard. It took years to get that thread through to the company to get them to give away that or give back that license. Do you see that aspect of Bayh-Dole being implemented much in terms of exclusive license, where it's clear that that's not actually being used, and then the university's in a position of having to try and claw back the ability to get it out?
Yeah, no, that's a great question, and it's definitely a real issue. This basically shelving a technology after purporting to be, oh, we're the perfect company to take this, improvement or whatever, and then they want to just want to keep their existing product. I have not heard much, if anything, of like trying to leverage Bayh-Dole to kind of claw back. That's an interesting thing to see if there's provisions relating to that would provide at least leverage to do that. The way that I've always thought about it is and I'm sure you have as well, is to try to deal with that through annual payments and kind of benchmark, benchmarks that the primary one is an annual payment to incentivize that company to just keep working on doing what they're supposed to do and purported to do. And but that's a really good question. I could see myself kind of diving into Bayh-Dole more directly and determining that, because it's a tragedy actually, to go through all the trouble of basically licensing, a new technology and then have a quote unquote partner basically not, take the baton and actually get it to market.
We used to see this at the front end. I haven't seen it recently, but years ago at Pitt, the argument would be, well, this startup that you want to form seems a bit too fledgling to us, and instead of giving the license to you, we've got Medtronic over here. They're clearly in a better position to commercialize this, and according to Bayh-Dole, we have the obligation to go with the more likely public benefit. So therefore, we're going to license to big company instead of startup company. Although nowadays that's not as common. Like I said, it used to be a little bit more common.
And there is the statutory requirement to favor at some level small companies as well, and that's something that we've used. That is in the Bayh-Dole. You have to give a preference to small companies, and it could counterbalance, certainly any. I get the arguments about the larger company, but my experience is, here at Davis and then Case Western a long time ago, is that it's, at least with the staff that I work with, we use that reasoning to support, the logic of going with startups, actually, even though they're obviously more risky in a certain sense, but that's it's in the law, and we're trying to help, especially if you have a faculty member and a team that really is interested in doing that. Then in my mind, those are the elements you need, and then you have the law behind it to actually back you up.
Thank you.
Okay, barring any more questions, I'd like to.
See, Kay Fuller.
There's a hand raised, yes.
All right, go for it. Go for it.
Hi, thank you very much. What a great talk. I'm just curious if I understood you correctly, that when an invention is disclosed to tech transfer, that it needs to be submitted within 2 months?
No. The 2 months is once it's submitted, whenever it's submitted, the tech transfer office has 2 months to inform the federal government if it's going to exercise its right to kind of take on that patent.
So what is that? What is that known as? That's not the same as a provisional?
No, it's not. Well, it's just, I don't know if it has its own name. The rule that of Bayh-Dole saying that once an investigator submits a invention disclosure, that triggers a time period that the university has to inform the federal government. A provisional basically is another mechanism to get a patent application on file.
Right. That's what I thought. I was inferring that's what that meant. I'm glad to hear that's not That
No,
seems
no,
like
yeah,
a
that's-
ridiculous
that's a
amount
bit-
of
no,
time.
no.
Yeah, it's too short. Thank you. Thanks
Right,
for
yeah, that's too
clarifying.
short. No, I didn't mean to juxtapose those. Those are like separate things.
Thank you.
Great question. Great question. Thank you for that clarification. Okay, well, let me wait a minute, see if any other questions or thoughts? Okay, well, I want to thank Jan and Jim for these very insightful talks and for being our initial speakers of our hopefully seminar series. And thank you all for your attending, and have a great day. Okay.
Thank you very much.
All right, thanks a lot.
Bye-bye,
Thank
guys.
you. Bye-bye.
Bye-bye.